Hi guys, welcome back to part 2 of chapter 3, and this is going to overview protein synthesis and cell transport, cell membrane transport, and it's a lot of physiology in this section. I'm on page 109 where we're going to start reviewing about the nucleus and then look at some things that the nucleus can direct. Nucleus, usually the core, usually the largest cellular structure. It's the control center or the brain, meaning it can direct the synthesis of over 100,000 different proteins, including the enzymes that catalyze all those metabolic pathways. There's a sequence of nucleotides that has important genetic information. There's usually only one per cell, except for skeletal cells. Skeletal muscle cells have multiple nuclei, and mature red blood cells, part of their life cycle is to actually eject the nucleus. So the mature red blood cells have none. If you look at this picture on page 109, you can see that this kind of shows you how the nucleus directs some adjustments when there's a change in the extracellular fluid outside of the cell. So, going through this, any kind of change in the chemical composition or anything really, can have two things happen. If it's a molecule that's small enough to go through the membrane, it will go through the membrane, so it will diffuse. And that throng, that just caught my attention, that's supposed to be through. Other things that are too big or maybe too charged, that will just bind to membrane receptors and trigger a pathway. So some of the things, once the nucleus recognizes this, just kind of like our brain recognizes a part of the homeostatic mechanism that we talked about in chapter 1, there are some short-term adjustments. They can, the DNA in the nucleus can actually start to transcribe and translate enzyme activators or inactivators or enzymes themselves, more enzymes, less enzymes. Some long-term adjustments, they can change biochemical processes or change the physical structure of the cell, meaning they can actually make more membrane or, direct organelles and things like that. All right, the next section is all about the nucleus and the nuclear membrane and some of the more particular structures inside. The nuclear envelope or nuclear membrane surrounds the nucleus. It is a double membrane bound organelle, so there is some space in between. They do have pores, so there's some things that can come out of there. Remember, transcription and translation, the RNA actually leaves the nucleus through these pores. The nucleoplasm is kind of comparable to the cytoplasm. It's the fill inside the nucleus, and it contains fine filaments for structural support, but also ions, enzymes, nucleotides, and some RNA and DNA. The nucleoli is this concentrated area within the nucleus, and there is a lot of RNA and enzymes in that area, other proteins as well, that are assembling the RNA subunits, so the larger and the smaller subunit that are part of translation. These are very prominent in areas where there's a lot of proteins that are being manufactured, for example, the liver, nerve, and muscle cells. Inside the nucleus, the DNA is very, very tightly coiled. It's actually wrapped around some histone molecules, and they form these nucleosome packets here. Histones are a type of protein there. The chromatin is the loose chromosome. So if it's a non-dividing cell, it's chromatin. If it becomes tightly coiled and looks like that X, then it is called a chromosome. And that's in dividing cells. During cell division, they form these tight chromosomes, so the chromatin becomes really, really tightly coiled and doubled, held together at a centromere. And at there are some sections in here about later in the chapter about mitosis. So plus we're covering that in Latin. There are 23 pairs one set of pairs, one side of the pairs is from mother and the other side is from the father. And they are instructions, blueprints. There's also some DNA that's not read, that's not a part of anything. And so some of them we know about, some of those we don't. Protein synthesis. So 3 types of RNA, enzymes, and DNA are involved. If you look on page 112 and 113, and actually it will continue on as we talk about transcription and translation, it has some great pictures. Just to remind you, DNA is a double strand. It looks like a ladder. They are held, the 2 sides of the ladder are held together by hydrogen bonds between the nitrogenous bases. So the steps of the ladder, or the rungs of the ladder, are composed of a complementary pair of nitrogenous bases, so A's with T's and C's with G's. Apples grow on trees, carrots grow underground, and they are bonded by a hydrogen bond. And then it's tightly twists and coiled because DNA is slightly negative, so that's what helps it keep it coiled. Along the DNA, there are a set of 3 nitrogenous bases, a triplet code, and that will transcribe into a codon, which will translate into a single amino acid, and those amino acids come together to form a protein. A gene is a section of the DNA, a sequence of the DNA, and it will correspond with specific proteins. Here is one example of the genetic code. In biology, you might have had the codon charts. You can read it as a wheel, or sometimes it's a big chart where you know, the first base, the second base, and the third base, or the first base is in the middle, the second base is in the next, and then the third base, and you read the amino acid that way. Well, this just shows you the relationship between the DNA coding, the mRNA, the tRNA, and then the amino acid. Of course, there's many more of those. All right, so protein synthesis, gene activation. So the gene has to be turned on, okay, so that it's available to transcribe and translate. And then it has to be accessible. So DNA strand has to uncoil and separate so that it is accessible for some of these enzymes to come in and transcribe. There's an enzyme that comes through and basically constructs a single strand of messenger RNA. So, and that's on the rules of complementary base pairing, but except for that T in mRNA, it's going to be a U. The mRNA then is going to leave the nucleus through the pores. In the cytoplasm, the 2 ribosome subunits come together, and the mRNA attaches to these 2 subunits as they're coming together. And then we have these anti-codon tRNA molecules. The anti-codon is the matching, that complement to the codon here. And so that tRNA, because of that anti-codon matching here, that refers to a specific amino acid. And so that amino acid, eventually what you'll see here are bonds, peptide bonds that are formed between them. And that's a fully, that's a polypeptide chain. And then you'll get several of those together, which will be a fully fledged protein. And this is the overview, which is also on page 113 in your textbook. So the kind of the flow of synthesis here, the DNA determines the sequence of mRNA codons. The mRNA codons determines the sequence of the tRNAs. The tRNA determines the sequence of the amino acids of the polypeptide chain. So you can kind of see the flow from DNA to mRNA to protein. The next part of the process is going to go through, or the next part of the chapter is going to go through these in detail. If you have your phone and you have a QR reader, or you just take a picture, you see all of these little QR codes here in the corner. There's also, there may be some on the back of yours. There's not on the back of mine because this is a this is an instructor copy. So some of you might have a QR code in the back of the book. You might want to check that. Some of it is the smart art, like within the chapter is the smart art to help you with these pictures. But the one on the back of the book, if you have one, is flashcards. I don't know if I told you that I used that the first day. I don't know why some of them came with it and some of them didn't. So if you don't have one, let me know and we'll just find a textbook with it on it next time you're in class and you can snap the QR code. All right, transcription is to take, it's that first part of that flow, to take the DNA and make an RNA strand. The steps of transcription, gene activation, it occurs at a segment, just a little part of the gene. And so that only one side of it's going to be a template in order to make the mRNA. RNA polymerase is that enzyme, and that's going to bind to a specific region of the gene, and it's going to start to make an mRNA strand. And it's going to continue to grow until it hits a code that's actually going to shut it off, stop code. And then transcription ends. The mRNA detaches, and then the DNA kind of rebuilds itself. So this mRNA that's made during transcription isn't necessarily what is going into the nucleus. It has to go through some processing, some editing. A lot of textbooks actually call it mRNA editing, and it's a second step in between transcription and translation. So before it leaves the nucleus, it requires some additional processing. The introns are removed because these are non-coding, so they're not really important. And the remaining exons are spliced together. So if you can see how it's edited here, the B is an intron and it's removed, and so it becomes A to C. There's also some things that are put on the front end and the back end of the mRNA to kind of protect it from the cell, and then it leaves the nucleus. The next phase is translation, and this occurs in 3 steps: initiation, elongation, and termination. Initiation, so you've got these ribosomal subunits and the mRNA that's all binding together. The first site is called the P site. The tRNA binds to this P site, and it starts to starts the initiation phase. And then, so the codons and the anticodons bond, and that's bringing the different amino acids together. Here you can see the small ribosomal units and all of us coming together. So I like the E, I like to say that's the exit because that's the site where, you know, tRNA is actually exiting. And then the P is kind of like the parking where they are bonding, and, you know, you're starting to put that polypeptide bond, or excuse me, just peptide bond between the 2 amino acids. It's growing. And then the A side, I don't have anything for that, sorry. But it's kind of like this green mRNA strand is a rope, and you're pulling the rope. So all of these tRNAs are going to shift. That one exits, the A one becomes the P one, and then another one comes into the A spot. The second step is elongation, and this is just as the amino acid chain grows. So here you see you're pulling that green mRNA, you're growing the strand. Termination, there's a stop codon on the mRNA. There's actually a few that are the stop codons. And so that kind of triggers everything to be released and to be reused. So the overview is on page 116 and 117. And this would be a really good point to actually stop and watch this video, stop and look at page 118 where the review for this section is. Just kind of take it all in, remember what you can remember, learn new things, that kind of stuff. Next, we're going to talk about transport in this section. So they're kind of the 3 levels of permeability. If it's freely permeable, any substance can pass. If it's selectively permeable, only some substances can pass. And when it's impermeable, no substance can pass. So our plasma membrane cells of any living thing are selectively permeable. The permeability of our membrane, you can see this would be the center one here, where some lipids are allowed in. Remember, this is a phospholipid bilayer, so lipids are going to be able to pass right through. This is kind of misleading. Some water is able to diffuse through, but there are also these channels, these aquaporin proteins that allow other water molecules to enter. So it's kind of like some water molecules can get through and some can't. And then these larger like proteins and carbohydrates, they cannot get in, and these ions are charged, so they cannot get in. The materials some characteristics of materials, the size of it, the smaller molecules can certainly pass through, the shape, the solubility, the electrical charge, and some other factors. So that's why some of them can get in and some of them can't. The cell membrane characteristics, we have lipids and proteins present, which means that some of these characteristics can pass and some of them cannot. And then how it's arranged. So it was arranged with the two ends of the phospholipids, the two tails facing each other, and then the two heads on the inner and outer surface. So there's two kinds of transport, and we're going to actually do some things in lab with membrane transport, and whether you're in person or you're doing the Mastering E&P. But the passive, there's no ATP required, and so that's why it's known as passive. And then so that's diffusion and carrier-mediated transport. Some of that is passive. And then active vesicular transport, which means usually the membrane is moving, or carrier-mediated, and that's usually active transport, what we know of as active transport. So passive transport, diffusion. Diffusion, we'll first talk about concentration gradient. A concentration gradient, any kind of gradient, is when there's a difference in something. And a concentration gradient is when there's a difference in the concentration of the molecule. So a concentration gradient, I have 17 molecules over here and 7 over here. There's a difference, so there's a gradient. There's a higher level over here and a lower level over here. And so that's all we're talking about. Sometimes in between that concentration gradient, there's a membrane. And so that's why we talk about diffusion and osmosis and active transport. Diffusion in air and water is slow, but that's okay. It is what it is. It's also dependent on the strength of the concentration gradient. Movement of waters and solutes across the membrane. Lipids and lipid-soluble molecules can pass right through. There are some channels, water and small water-soluble molecules, and some ions can go through a membrane channel. Some use carrier molecules to get them across. It is just what it sounds like. And this just shows you some of those, how they would diffuse across the membrane, or maybe they go across a protein, or maybe they are, there's a, I'm out of control. The carrier protein. So it just depends. This is passive transport, so no energy. And here's some factors that influence how fast the diffusion occurs. Temperature is a good one. The strength of the concentration gradient, the size, the distance, and if there's electrical forces. Osmosis is the one that is most related to anatomy, because there has to be a balanced flow of water. Osmotic flow is the movement of water driven by this process, and of course osmosis is the diffusion of water. The osmotic pressure is an indication of how much force the pure water moving into a solution with a higher solute concentration. So I always said in my biology classes that salt sucks. So wherever there's higher solute, or higher, and most of the time we use salt as an example, that's where the water is going to move into. And osmotic pressure is just the pressure involved in that movement. Hydrostatic pressure is the pressure of the fluid itself. Here, this is a I don't know why they still do these view things. It's easier to understand when you're doing when you're looking at like a cell and how it's affected the cell. But if you have a selectively permeable membrane, it's permeable to the water, but not to the solute. So wherever there's more solute, and it looks like there's more solute over here, the water is going to flow, okay? But when you apply a force, you can actually force the membrane, the water across the membrane. This is just some, you can actually calculate the concentrations and calculate the force. Osmolarity is the total solute concentration in solution. You may have seen the word molarity when you studied chemistry. It's similar. Tonicity is how the solution is going to affect the cell. And when you talk about tonicity, usually you're starting to talk about isotonic, hypotonic, and hypertonic. So isotonic means the same. It means the net flow of water is equal in, equal out. This is ideal for our cells, animal cells. And you can see here the solute's not moving, but the water molecules are in and out. And it looks like a healthy kind of disc-like red blood cell. Hypotonic with the O, that's a swelling. So that means that there's more concentrated solute inside of the cell, and so water is rushing in. And this is called, if it's a red blood cell, this is called a hemolysis. Hemo meaning blood, and lysis meaning to burst or break, they say loosening. So what's going to happen is this is going to swell like an O in hypotonic and then burst. Hypertonic is the opposite, when there's more solute out here in the solution. And so water from inside of the cell is going to have a net flow out, and it's going to cause the cell to shrink and shrivel, and this is called crenation. And there's the three kind of side by side. This is what your healthy red blood cell should look like, the red one over here. So this is important because sometimes patients need fluid, either saline or blood, whatever. But you have to match the osmolarity of that, that internal fluid, okay? So going too high, of course, would cause the cells, high solute, that is, would cause the cells to crenate, and having a low would cause the cells to hemolyze, excuse me. So normal saline is 0.9% of NaCl. That's kind of what our cells are normally isotonic with. The last part of this section is about some of the other types of transport. Carrier transport involves a protein, and that can get large molecules across. There's some different examples. Facilitated diffusion is a type of diffusion where there is a carrier molecule, but we're still going from high to low concentration. So there might be some ATP required, but it's still considered passive. Active transport will actively pump, instead of from high to low, from low to high. And the sodium-potassium exchange pump is a very good example. Okay, this is important to, and not just this one, but active transport ion pumps are very important when we're talking about muscle contraction or nerve conduction, things like that. So this secondary active transport is usually a pair. It's a lot of the examples I've heard of is it's a movement for two substances that follows the concentration gradient that goes down. And here's another video, so this might be a good point to pause. The last part of this section is about vesicular transport, and this is endocytosis and exocytosis. Basically, when the membrane is moving itself. So endocytosis is when extracellular substances are actually engulfed by the membrane. Exocytosis is when they're secreted or they're removed, like in wastes. The receptor-mediated endocytosis is a type of endocytosis, but if you see, it involves these receptors in the plasma membrane. And so things will bind to the receptors, okay? I think this is like that. Yeah. And then the plasma membrane then triggers the actual folding. Eventually that will become a vesicle within the cell itself. And there's some details that really aren't important. Okay, so this is the overall of receptor mediated, and you can see the receptors here, where then there's a ligand or it's a molecule that binds to those receptors and triggers the endocytosis process. The lysosome will often fuse with that and then sort of take what it needs, you know, take those ligands, and then the pieces of the plasma membrane will actually return to the plasma membrane. Pinocytosis is endocytosis where it's liquid, and phagocytosis is where it's not liquid, and usually that involves some kind of pathogen like a bacteria or virus. Exocytosis, opposite of endocytosis, you're secreting things out or you're trying to get rid of wastes. Okay guys, that's the end of this section on page 128. There is a really great flow chart, so you might want to consider putting that in your notes just to kind of summarize all the types of transport. If you have any questions, email me. Otherwise, on to part 3.